Now let's come to the whole exome sequencing. So now if you see, there is a catch, when I mentioned the genome sequencing, when I mentioned the sequencing, I did not mention about the exome. So ideally it would be, if it is from DNA, it would be genome. What is the difference between exome and genome? I would say I will explain entire procedure once, and I will explain the difference also. Here, you isolate the DNA from the sample. Okay, as I mentioned, let's say you have a blood sample or saliva, whatever. You isolate the DNA and you do the fragments same way. But here, the amplification during the amplification, you enrich only the exome. The exome. How do you do that? For that, the companies provide specific kits, like for example, Twist. You can note down this Twist capturing kit and Roche capturing kit. So they come Twist comprehensive kit, and like this same similarly Roche also. So you get the kit, you use and enrich only the exonic region or exome of the genome. So that is amplified, and only the exome is sequenced. And this is data analysis. I will explain this in detail.
Could you repeat, please, that the kit name?
Yeah, Twist comprehensive exome kit, exome target enrichment kits. Target enrichment kits, many are there in market. One of them is Twist comprehensive. And also, I guess now V2 is there. I'm not very sure. V2 is there currently. And also Roche. I guess Roche is also doing good as per my last observation. Okay. Now, the most important and interesting part of our workshop is data processing. Now, I want you all to imagine, now we have reads from the sequencer. Let's stick to the again Illumina. Now we have reads of 150 bases. Okay, so we have a bundle of reads like this. Okay, so these reads at first they are also in fast queue format. So what is fast queue, and all we will see tomorrow in tomorrow's session. In short, fast queue is a faster sequence, but a quality score is attached to, for each base. So what is quality score? What is correct quality score? Everything we will see in the tomorrow session. So now we have the raw rates. The first thing that we do once we have the raw rates is the quality checking. So we check the quality of the read, and also trimming is done. Why trimming is done? Mm, any question? Okay. So quality checking and trimming is the first step that is done once we have the raw rates. So now the trims are, sorry, the reeds are trimmed, and if there is any adapter contamination, or if the base quality is not good, that kind of reeds or the part of the reed is deleted. We have a clean reed. Let's assume we have a clean reed. Now, again, reeds are this way. That has to be aligned to the some reference genome. I will again talk about the reference genome in detail tomorrow. But we have different reference genome like HG19 or HG38 for the human I'm talking about. So HG19 or HG38 are mostly in use. I would say HG38 is mostly in use. Okay, so now reads are aligned to the HG38 reference genome. So map to the reference genome is done. This is fine. Now we have reads aligned to the reference genome, but some reads might be, since the amplification procedure is involved in the wet lab, so you might have more than one read, a duplicated read at the same position. I mean to say a duplicated read. It's not actually a different read, it's a duplicate of the same read. Again, this also we will see in detail in our future sessions. So marking these duplicated reads is very essential. So the next step is we mark the duplicate reads, and also we do the base recalibration. So what is this base recalibration? So initially we get some quality score assigned to the each base in the read. So that has to be recalibrated. Uh, I will explain the reason. Then once this is done, we get a BAM file. So so far we have a fast queue for after alignment. We get a aligned BAM file, binary aligned mapped read. This is a file format. So we get reads in BAM format. fall after that, what happens? Now we have reads in recalibrated reads in BAM format. Then variant calling is done. So what is variant calling? So now a tool, for example, let's say GATK haplotype caller. What it does, it checks for it checks locally for each wherever there is a variant or there is a difference from the reference genome, it does the local realignment and calls the variant. So uh, so this way we will be having a VCF format, VCF format with a number of variants. This is further filtered by different methods and then annotated. uh, Currently you may not understand, but this is what we are going to understand throughout our this course, this session, I mean this workshop. But I just want you to introduce to certain terminology. What is fast queue, alignment, mark duplicate, base recalibration, then BAM format, what is VCF. I mean, you don't need to know what is VCF also, but the terminologies I want you to introduce to. Then annotation. That's it. If you just remember this terminology, in future we are going to discuss about them in detail.